what if the exact same mineral that builds your skeleton is, secretly rusting at your cells from the inside?
Yeah, it's a pretty wild concept to wrap your head around.
Right. I mean, we've been told our whole lives, drink a big glass of milk, get your calcium for strong bones and healthy teeth. You picture this inert, chalky building block just solidifying your frame.
Exactly, just acting like scaffolding.
But what we're unpacking in today's deep dive turns that entirely upside down. It turns out a microscopic calcium leak might actually be like the primary engine of human aging.
It really is a complete paradigm shift from how we usually think about biology. So we're pulling from this fascinating July 2026 report by lifespan.io, which covers a breakthrough study published in Nature Communications by a team of Chinese scientists.
And the reason the study is turning so many heads is that it treats the aging process not as some mystical, unavoidable decay, but literally as a mechanical failure.
Right, a mechanical breakdown that you can actually trace.
Exactly. So our mission for you today is to trace this microscopic plumbing leak inside your cells, see exactly how it drives both accelerated and natural aging, and then explore how an old, very common antidepressant medication might be the unexpected wrench we need to fix it.
Sounds like a plan.
But to understand the fix, we have to look at the crime scene first. Let's talk about what calcium is actually doing inside a cell, because it's definitely not acting like bone scaffolding down there.
Oh, far from it. Inside a single cell, calcium ions, the biochemists write them as K2+, they function as the ultimate biological switches.
Like electrical switches?
Basically, yeah. A cell uses the rapid rise and fall of calcium levels in its internal fluid to communicate, to trigger muscle contractions, to release neurotransmitters.
To turn genetic pathways on or off, right?
Exactly. It is this incredibly fast, highly sophisticated signaling system. So maintaining that balance, what we call calcium homeostasis, is literally a matter of life and death for the cell.
Okay, so rather than bricks in a structural wall, we should be thinking of calcium as, the high voltage electricity in a city's power grid.
Yes, that's a great way to look at it. And just like a city's power grid, a blackout or a surge causes catastrophic problems. Researchers have known for decades that calcium dysregulation is linked to major age-related diseases.
Right, like we see it in heart failure.
Hypertension, age-related muscle loss, and even Alzheimer's. The calcium balance is just visibly broken in aging cells.
But the missing piece of the puzzle, though, was causality, right?
Yeah.
Because seeing a broken power grid in a decaying city doesn't tell you if the broken grid caused the decay or if the decay broke the grid.
Which is exactly the mystery these researchers set out to solve. They needed to trace the concrete molecular chain of events from start to finish. And to do that, they couldn't just watch normal, naturally aging mice.
Because natural aging is just a slow, messy grind.
Exactly. The signals are way too faint. They needed to turn the volume up, essentially. So they used a model of Hutchinson-Gilford progeria syndrome.
Progeria. That's the rare genetic disease that causes children to age at an incredibly accelerated, tragic rate.
It is, yeah. By using mice engineered with a progeroid mutation, scientists can basically hit fast forward on the aging tape. These mice are only 12 weeks old chronologically, but biologically, their tissues are elderly. Their organs are already failing.
Yeah.
So the researchers took lung tissue from these fast-aging mice and ran a technique called unbiased proteomics.
Unbiased proteomics. So taking an inventory of the proteins.
A completely blind inventory, yeah. You don't look for a specific suspect. You just cast a wide net to see which proteins are being overproduced or underproduced compared to a healthy young mouse.
And what did they find?
The results were glaring. The entire calcium signaling pathway was massively overactive in these fast-aging tissues. The cell's internal fluid, the cytoplasm, was just absolutely flooded with calcium.
But where is it all coming from? If the cell is flooded, there has to be a source of the leak, right?
Right. So they had to measure the different compartments inside the cell. They checked the mitochondria first.
The powerhouses.
Yep. And they hold some calcium, but their levels were totally stable, so the leak wasn't there. Then they checked a massive folded structure inside the cell called the endoplasmic reticulum, or the ER.
Okay.
And when they measured the ER, the calcium levels were dropping fast.
Oh, wow. Okay, I want to visualize this for anyone listening, because I like to picture the cell as a house.
Sure.
So the nucleus in the center is the home office, where the master DNA blueprints are kept safe under lock and key. The mitochondria are the fuse box, or the cytoplasm, the main fluid, is the basement. And the ER, this structure you just mentioned, acts as the cell's hot water heater.
Right, it stores a huge pressurized reservoir of calcium to be released only when needed.
Exactly. So that analogy fits the data perfectly. The basement is flooded and the water heater is emptying out.
Yeah, that's exactly what's happening. And the researchers tracked the source of the flood to a very specific release valve on the ER, a protein structure known as the IP3R channel.
The IP3R channel.
Under normal conditions, this valve opens for a split second, lets a tiny puff of calcium out to signal a process, and just snaps shut. But in these aging cells, the IP3R valve was stuck wide open.
So it's just blasting calcium continuously into the basement.
Yes, and this isn't just harmless water. A calcium flood inside a cell is highly toxic.
Right, it creates a domino effect.
It does. The researchers noticed that this constant flooding of calcium was strongly associated with a massive spike in one particular incredibly destructive protein. It's called S100A6.
S100A6. Okay, that is going to be the central villain of this story, isn't it?
Oh, definitely the main culprit. They found it elevated everywhere they looked. It was saturating the heart, the lungs, the skin, the muscles of these fast-aging mice.
It was just everywhere.
Everywhere. And they even cross-referenced this by looking at human skin cells donated by patients with progeria, and the exact same signature was there. Huge toxic amounts of S1000A6 piling up in that flooded cytoplasm.
Okay, so we have a stuck valve, a flooded basement, and a massive buildup of this S100A6 protein. The next logical step is, connecting that specific protein to the physical reality of aging.
Right.
How do we get from a protein floating in a cell to, wrinkling skin, fragile bones, and a failing heart?
Well, you have to find out what S100A6 is attacking, because proteins don't just float around aimlessly, they interact with other proteins.
Okay, so who is it interacting with?
The researchers used mass spectrometry, which is this tool that lets you see which proteins are physically binding to each other. They basically baited a hook with S100A6 to see what it would catch.
Oh, yeah.
And they found it was grabbing onto a protein called PARP1.
Okay, PARP1. That's one of the most critical maintenance workers in the entire cell, isn't it?
It really is. It's the cell's master mechanic for DNA. I mean, your DNA is constantly under assault.
Just from normal life, right?
Yeah, just existing, metabolizing oxygen, stepping out into the sunlight. It all causes physical breaks in your genetic code every single day. PRRP1 is the enzyme that rushes to the site of the damage, repairs the break, and keeps the code intact.
Wait, I need to clarify how this attack works, though. When this S100A6 villain binds to the PRP1 mechanic, is it destroying the original instructions to make the mechanic, or is it just destroying the finished worker once it arrives on the job?
That's a great question.
Are we burning the blueprints, or are we throwing the mechanic in the trash?
The researchers asked that exact same thing. So they measured the messenger RNA, the genetic orders that the nucleus sends out to manufacture PRP1.
And what did they see?
Those mRNA levels were perfectly normal. The nucleus is wide awake, detecting DNA damage, and frantically printing out orders like, we need more PRP1 mechanics out here.
So the cell's factories are successfully building the enzymes.
Yes. The blueprint is completely fine. But the moment the freshly manufactured PRP1 enzyme steps out into that flooded cytoplasm, the excess S100A6 proteins just swarm it.
Oh, wow.
They bind to it and trigger a process that degrades the mechanic before it can ever reach the nucleus to do its job. It's what we call a form of post-translational regulation.
Which means the cell is just churning out repairmen, but they are all being basically assassinated in the flooded basement before they can fix the DNA. That has to lead to immediate catastrophic failure for the cell.
It does. Without PRIP, the daily routine DNA breaks just don't get fixed. They compound. The researchers could see this by tracking specific molecular distress signals.
Flashing alarms, basically.
Exactly. Chemical alarms that indicate severe, unrepairable physical breaks in the DNA strands. They also saw a huge spike in signals that light up when a cell is forced into senescence.
Right, and senescence is essentially when a cell becomes so damaged that it permanently retires, right? It just stops dividing.
Yes, but it doesn't quietly retire. A senescent cell becomes a toxic zombie. And the reason it becomes toxic is directly tied to those unrepaired DNA breaks we just talked about.
Because the DNA is just too far gone.
Right. When DNA sustains enough severe damage, pieces of it physically snap off, and these broken fragments actually leak out of the protective nucleus and escape into the main fluid of the cell.
So the basement is now full of broken DNA fragments, and the cell's immune sensors absolutely panic when they see that, don't they?
Oh, complete panic. For a very ancient evolutionary reason, actually. To a cell's internal sensors, loose DNA floating in the cytoplasm looks exactly like a viral infection.
Wait, really? Just because it's loose DNA?
Yeah, because viruses reproduce by injecting their own genetic material into the main body of a cell to hijack its machinery. When the cell detects its own escaped DNA fragments, it can't tell the difference. It just assumes it's been invaded by a virus.
So it trips the alarm system.
An incredibly powerful ancient alarm system, the CGS stinging pathway, that triggers massive cellular inflammation. The cell is basically trying to burn out what it thinks is a viral invader, but the invader is just its own shattered genetic code.
It is burning down its own house. The tragedy of that mechanism is incredible. Let's trace this entire chain reaction from the beginning so you can see the elegance of what these researchers uncovered.
Sure.
Step 1, the release valve on the water heater gets stuck open. Step 2, calcium floods the cell, triggering a massive spike in the S100A6 protein. Step 3, that protein swarms and degrades PARP1, our master DNA mechanic.
Yep, spot on.
Step 4, without repairs, the DNA shatters and fragments leak out of the nucleus. And finally, step 5. The cell mistakes its own broken DNA for a virus and triggers chronic severe inflammation.
That sequence from leak to protein to lost mechanic to broken DNA to inflammation, that is the fundamental engine of cellular aging they managed to map out.
You know, seeing it laid out sequentially like that actually makes me optimistic. Because if we know exactly where the chain reaction starts, that stuck valve on the ER, doesn't that give us a very clear physical target to drug? Can't we just find a chemical that plugs the leak?
Well, the researchers tried the most direct, aggressive route first. They used an experimental chemical compound known to forcefully block that specific calcium valve.
Did it work?
It achieved the mechanical goal. It shut the leak. Cytoplasmic calcium dropped, the toxic S100A6 protein plummeted, and the PRP1 mechanics returned to the job. It even extended the median survival of the fast-aging mice by about 14% and helped them maintain their body weight.
Okay, extending lifespan by 14% is a pretty solid proof of concept. But I'm guessing a direct, forceful block of a crucial cellular valve comes with some severe collateral damage.
It really did. The drug caused severe, constant tremors in the mice. It's a blunt instrument, you know.
Yeah.
You cannot forcefully padlock a valve that the cell occasionally needs to function normally.
Right.
If you were developing a long-term therapy to slow down aging in humans, a drug that gives the patient uncontrollable tremors is a complete non-starter.
Right.
Quality of life has to be prioritized alongside length of life.
Right. They need a way to finesse the valve shut without breaking the rest of the machinery.
Yeah.
And this is where the lateral thinking of this scientific team just really shines.
Oh, absolutely.
They pivot away from these blunt experimental chemicals and start looking for something much more tolerable, like finding out your car's radio tuner can somehow patch a leak in your fuel line. And they find the solution in a drug called mianserin.
Yeah, mianserin is a very old, very well-documented antidepressant medication. It's been used for decades.
But how does an old psychiatric drug designed to affect mood in the brain reach it down and fix a microscopic plumbing leak on a cell's internal water heater? I mean, that sounds crazy.
It comes down to a really strange quirk of evolutionary biology. These internal cellular valves aren't just acting on their own, you know? They're listening to signals from the outside.
Yeah.
Mianserin's primary job, the reason it was prescribed as an antidepressant, is to block certain serotonin receptors.
The receptors that process the happiness chemical, basically.
Yes. But those exact same serotonin receptors don't just exist in the brain. They are present on the surface of these cells too, and they are directly wired to the IP3R calcium valve.
Oh, I see.
Serotonin signaling is one of the things that tells that valve to open. By applying mianserin and blocking the serotonin receptor, the researchers inadvertently cut the signal that was keeping the valve stuck open.
So without the constant signal, the valve naturally relaxes and closes. The leak just stops.
Exactly. They used a psychiatric drug to quiet the signal and let the valve reset itself.
That's incredible. So what happened when they gave this antidepressant to the fast-aging progeroid mice?
They started treating the mice at 4 weeks of age. And where the blunt chemical extended median survival by 14%, mianserin extended their median survival by roughly 30%.
30%. That is a massive jump.
It is, and the health span improvements were even more striking than the lifespan numbers.
Really? Like what?
The treated mice maintained healthier body weight. Their physical appearance was drastically better. When the researchers tested their cardiac, pulmonary, and muscle functions, they found significant preservation of tissue health.
So they were physically younger.
Much younger. And at the molecular level, they proved that S100A6 had dropped and the DNA repair mechanics were back on the job.
But how do they know for sure that the antidepressant didn't just accidentally trigger some other unknown survival pathway? I mean, biology is incredibly messy.
Right, that requires a validation test. So they took a group of the treated mice that were receiving the antidepressant and showing all these anti-aging benefits, and they artificially forced calcium back into their cells using a totally separate mechanism.
Okay, kind of overriding the drug.
Exactly. And the moment they flooded the cells with calcium again, all the anti-aging benefits of the mianserin completely vanished. The mice deteriorated rapidly.
Wow.
This proved definitively that the antidepressant was working specifically by fixing the calcium leak.
Okay, if you are listening to this right now, the obvious question staring us in the face is translation.
Of course.
Curing accelerated aging in a mouse engineered to have a rare genetic disease is an incredible feat.
Mm-hmm.
But, most of us do not have progeria. Does any of this sequence apply to the slow, natural, everyday aging that you and I actually experience?
The researchers knew they couldn't publish without answering that exact question. Progeria is a great magnifying glass, but you still have to prove that what you see under the glass exists in the real world.
Right.
So they went hunting for our main villain, that S100A6 protein in naturally aged subjects, and they found it. It was highly elevated in skin cells taken from very old human subjects. They also found it saturating the tissues of naturally aged elderly rats.
But does it spike anytime a cell is stressed, or is it unique to getting older? Because that's a big distinction.
They tested that, and the nuance there is crucial. If you subject a young, healthy cell to a sudden, acute stress, like blasting it with radiation or a harsh chemical, the cell will panic and go into senescence, but the S100A6 levels actually drop.
Oh, interesting. So it's not just a generic stress response.
No, that proves this protein isn't a generic panic button. A flooded cytoplasm full of S100A6 is a specific, unique molecular signature of chronic physiological aging, the slow grind of time.
Okay, since the mechanism holds up in natural aging, they had to run the ultimate test, right? Trying the drug on naturally old mice.
Yep, they set up a 4-month trial. They took a cohort of wild-type mice that were already in the later stages of natural life, and they gave them either saline as a placebo or mianserin every other day.
And what happened to the naturally old mice?
Looking at median survival, the mice treated with the antidepressant lived 17.5% longer.
A 17.5% extension when starting the intervention late in life is profound. And I imagine the physical improvements were just as visible as they were in the progeria model.
They were visually distinct, yeah. The treated mice had glossier fur, they maintained larger body sizes, and avoided that frail wasting that usually happens at that age.
That's amazing.
They had significantly less spinal curvature, that hunched posture known as kyphosis. They showed far less osteoporosis in their leg bones, and their overall locomotion and movement were vastly superior to the placebo group.
Okay, let's pause and play the skeptic here for a second. Glossier fur, stronger bones, a straighter spine, and nearly 20% more lifespan from a cheap, off-patent antidepressant. When the results sound this miraculous, we have to look for the fine print. What is the catch here?
Yeah, you always have to look at the fine print. There are 3 major caveats that anyone in geroscience would point out immediately with this study.
Okay, what's the first one?
The first is the scale of the natural aging experiment. The cohort size was very small, only 7 or 8 mice per group. In rigorous longevity research, you really need dozens, ideally hundreds, of subjects to smooth out statistical noise.
Right, because small sample sizes can easily generate flukes. What is the second caveat?
The cohort was entirely male. Biological sex dictates massive differences in aging pathways, hormone profiles, and how drugs are metabolized by the liver.
Absolutely.
You can never claim a compound is a universal anti-aging intervention until you have robust data proving it works equally well in female models.
Right. And the third caveat is something I find fascinating because it highlights just how difficult it is to study longevity.
Yeah.
It involves the control group, right?
Yes. It's a known pitfall that researchers refer to as the 900-day rule. In this specific experiment, the control group, the mice getting the saltwater placebo, were a standard laboratory strain called black 6 mice. But their median lifespan in the study was only 823 days, with a maximum of 869 days.
Why is that specific number a red flag?
Because a healthy, well-housed black 6 mouse should easily live past 900 days. For whatever reason, the control group in this facility died unusually young.
Oh, I see.
And when your baseline group dies prematurely, any intervention you give to the test group is going to look like it provided a massive, perhaps artificial, lifespan extension.
So the antidepressant might not have pushed the test mice into, like, unnatural longevity. It might just be that the control group had a suspiciously short lifespan, making the gap look wider than it really is.
That is exactly the risk. However, you have to look at the totality of the data here. Even if we put an asterisk next to the exact percentage of lifespan extension, the healthspan data remains incredibly robust.
Because a short-lived control group doesn't magically cure osteoporosis in the test group.
Exactly. A wonky control baseline doesn't reverse spinal curvature, or restore muscle function, or bring back glossy fur. The sheer physical vitality of the treated mice, alongside the molecular proof that the calcium leak was sealed and the DNA repairmen were back to work, it provides deep reassurance. The drug unquestionably slowed the physical decay of aging in those tissues.
It grounds the math in physical reality. Let's pull all these threads together then. We started this deep dive, looking at a mundane glass of milk, picturing calcium as just a static building block. But instead, we zoomed into the high-voltage electrical grid of the cell. We found a microscopic leaky valve on the cell's water heater.
Right, and we watched that calcium flood trigger an assassin protein that destroyed our master DNA mechanics.
We watched the unrepaired DNA shatter leak out and trick the cell into setting its own house on fire through massive inflammation.
It's a brutal chain of events.
And the most incredible part of this entire journey is the fix. The solution wasn't some futuristic billion-dollar synthetic compound engineered in a vacuum. It was mianserin.
Yep.
An old, widely available antidepressant that just happens to silence the serotonin receptors that were keeping that cellular valve stuck open. I mean, this fundamentally changes how we view the future of human longevity.
It really does. It shows us that extending human health span might not require us to invent entirely new, impossibly expensive molecules. It might simply be a matter of mapping our own cellular engine with such incredible precision that we realize we already possess the tools to fix it. It's almost the ultimate form of molecular recycling.
We're literally just learning how to read the manual, which leaves us with one final, very exciting idea to chew on.
Oh, for sure. If a standard everyday psychiatric drug can unknowingly patch a microscopic plumbing leak that drives cellular aging, it forces you to wonder, what other age-reversing keys are currently sitting on the shelves of your neighborhood pharmacy, just quietly waiting for us to discover their hidden mechanical uses?
The medicine cabinet of tomorrow might be exactly the same as the one we have today. We just won't be reading the labels the same way.
Exactly.
Thank you for joining us on this deep dive. Keep questioning the everyday things around you, and we will catch you next time.